Yeah, so today we're going to talk about Marvin Live at Sprint Bioscience and how Marvin Live can facilitate structure-based design for us. So just to set the scene, what we want to do is better molecules, and we want to make better molecules faster. And I would say this is one of the key challenges in preclinical drug discovery. So a few words about Sprint Bioscience. Sprint Bioscience, we are a drug discovery company focusing on cancer treatment. We're fairly small, we're 23 employees in total, and the company's been built up around disciplines that we think are important to be able to start and drive small molecule drug discovery projects in the preclinical phase. So, let's see, laser. We start by selecting target proteins that we think are relevant for treating cancer. And in this selection phase, technical feasibility is very important to us. We want only to use and to work on target proteins that can be crystallized so that we can use X-ray crystallography and structure-based design. So in the company we have protein science and biophysics to be able to do that in an efficient way. And in the next step we find hits. We use fragment-based techniques, fragment-based screening, to identify binders to our target protein. And then we take these fragments, these binders, and very easily we grow them and we optimize them into molecules that are ready to go into in vivo testing. So of course, the box medicinal chemistry encompass where a lot of people, that's where the largest part of the work is done. So in the company we have a medicinal chemistry team, we're 8 people, including computational chemistry, that's Jenny, and non-computational chemistry, that's, for example, me. So synthetic chemistry and medichem and project management. And compound management, that's me as well. But then, of course, we also have biochemistry and cell biology to have target-related assays so we can drive the project forward. But when it comes to ADME profiling, all the in vivo work, selectivity assays, we do that with collaborators and CROs. And as you can see on this slide, we don't have a box for IT. So in that respect, I'd like to go back to Ivan's slide from yesterday. What we want, what we need at Sprint Bioscience is all this powerful clockwork behind the scenes. But we want the iWatch. That's what we want. And that's partially why we're here today and talking about Marvin Live. So just coming back to here, what we will want to talk about today is we want to zoom in on the box here that we call medicinal chemistry. So driving medicinal chemistry plans, most of you have seen this type of slides before. What we want to do, all the compounds that we make, when we want to make better molecules and we want to make them faster. A better molecule is what drives the project forward. So we want to design a better molecule. So we have a hypothesis, what do we want to improve? We design a molecule, we make it, we test it in the appropriate assay, potency or permeability or whatever. We analyze the result and we go back to the design stage. So once again, zooming in, design. Like I said, we have a hypothesis. Can we improve potency by building into a pocket? Can we reduce improve metabolic stability by reducing lipophilicity? We need to populate that hypothesis. We need to populate that idea with compounds so we can test the hypothesis. And we can populate it in different ways. And today we will talk about, on this talk, on how we use Marvin Live to populate that, the idea. So what structures actually look at. And then we go to the evaluation stage. Jenny has a lot of really nice models that we can use. We have a lot of crystal structures, a lot of these things. But we also have really skilled chemists. So we always make sure that we have synthetically feasible ideas. So in the end, what molecule to actually make. So that's all fine. But how to make the ideas better and how to make them faster. So we asked ourselves roughly a year ago, what do we miss at the medchem team at Sprint Bioscience? Well, first of all, access to the 3D world for everyone. Of course, we have a lot of crystal structures. We can view them. Everyone in the company can view the crystal structures. We can overlay structures. But non-computational chemists, so if I didn't have any tools to actually be creative and design molecules in the 3D world. Even though these molecules are fairly flat, it's still just drawing them on a piece of paper, it's very 2D. So bringing the 3D world to everyone in the team, that was one of the things that we wanted to do. And the second thing is that we want people to be able to check their idea and make more ideas and learn and be more creative and create better ideas. Instead of handing a lot of post-its to Jenny, can you check these please? Because then the iteration will be very slow. If I can check this myself, I can make another idea that is even better, or I can make more ideas. And in the end, to improve the workflow, how do we actually keep track of all our ideas? How do we share them? In the end, we will have, on Jenny's desk, there will be a lot of post-its. And also, she's away, and then she comes with a new post-it, and she just gets one of these post-its with structures and a question mark, what to do with it. So this is what we wanted to improve. So the question is how to make better ideas using Marvin Live. Then I'll give this to Jenny.
Yes. How are we using Marvin Live to create these ideas? First, we just have a look at the mission. This is a pretend case, but it's very similar to what we do every day. We have a kinase inhibitor. We have the crystal structure. We see how it binds to the active site. An acceptor to the hinge. We have a phenyl in the selectivity pocket. But we also have major solubility problems. So we need to make some drastic changes to this molecule. We have a chemist who is drawing this benzpyrrosol in Marvin Live. And his question is, will this benzpyrrosol with the acceptor find the acceptor position to the hinge and at the same time find the selectivity pocket with the phenyl? So that's the question. Using the alignment in Marvin Live, we see that indeed, this looks like a very good idea. Also, as Daniel and Andras was mentioning, the phys-chem properties, the calculated properties, are also important for a medicinal chemist. So polar surface area, lipophilicity. We can have a look at those properties. And together with the alignment, we decide that, yes, this is a good idea. So then the chemist will save the idea as an SD file and as a report in PowerPoint. And he will send this to me for further evaluation in more sophisticated models. Here comes the SD file. So I'm very happy with post-its, but if you want to make a computational chemist really happy, you give them an SD file. So with the SD file, I can directly plug in these ideas into my models. Those could be dockings or QSAR models or whatever. And since I have also the question, with this core, can the phenyl reach the selectivity pocket and still maintain the acceptor to the hinge? With the question, I can more easily understand what the intentions of the chemists are. And I can look for that in my models. So I get the report file from Marvin Live. And I don't need to draw the structures myself. The only thing I need to do is to check the structures in my models. I add the heart on the compounds that I like, or my models like. I put the stop sign on the compounds that the chemist was also drawing. Now, actually, the isoquinoline core did not fit to our protein in my docking models. So I just put the stop sign on that molecule. And then at our chemistry team meetings, we will look at this report together, and obviously we will decide that, yeah, the middle idea, that's the good one. That's the one we will synthesize. And since this is a pretend dummy case, we can actually see from the PDB where all the literature crystal structures are deposited, that indeed Marvin Live could reproduce the true alignment of these two molecules to our kinase. That's very nice. So now we want to continue building on our new idea. The blue idea was very good. And actually our orange reference here is even larger than you saw before. So we have an amide that is growing from the core, and we want to do something similar on our new blue scaffold. We want to build in the same direction. So again, the chemist is drawing something in Marvin Live with a question. Does this phenyl fit as an amide isoster? And actually, you get a really bad alignment in Marvin Live this time. But as a chemist, you know that usually phenols, they can do kind of the same thing as an amide. You write in the task field a question to me. Does the phenyl fit as an amide isoster? I get a bad alignment in Marvin Live, but could you please double check? I will double check. And indeed, my more sophisticated models will recognize that that's an okay idea. I put the heart on it. We will synthesize it. And again, dummy case, we know the answer. Indeed, a phenyl and an amide can replace each other in this case. So to summarize, why better and faster using Marvin Live? We think that if a chemist or anyone sees this really nice alignment, they will be happy, they will be inspired, and they will draw even more analogs to this good idea. We will get more ideas, and better. And also, I think maybe writing the hypothesis in the task field will actually help the creator to focus on the question. I'm trying to reach the selectivity pocket, and when you write it, you get an immediate reply how it's going. You will focus on making better ideas, and I can focus on helping them answering this. I could even make little changes to their ideas to make this fit into the selectivity pocket better. I think the evaluation goes faster since I get the SD file. The SD file goes directly into my models. The only thing I need to do when I'm generating the report is to add my little hearts, my stop signs, and then we can discuss it together. And another important aspect on the faster part is that the more ideas we get, the higher the chances are that some of these ideas will be more synthetically feasible. So we can pick the ideas that kind of replies the same question, but they are faster to synthesize, which will drive this DMTA cycle faster. We have some enhancement suggestions. Better alignments. Here we see an example where the acceptors actually didn't align on top of each other as we expected. And we don't expect this to be like a Schrödinger quality alignment. We don't expect it and we don't want to pay for it. But still, if it can be improved and still be fast and still be cheap and super good, that's what we want. We were talking yesterday about the ability to add more references at the same time. So usually with different references, we can see how different references can expand into different parts of the active site. So we think we can increase the inspiration by looking at several references at the same time. And also, if you are able to align on several references at the same time, maybe your initial idea was intended to align on series 1, but actually it happens that it aligns better on series 2. That would be nice. Also, we would very much like to add the surface of the protein and all atoms of the protein. So again, we know that the alignments are purely ligand-based, and we're fine with that. We don't expect it to be a docking. But still, if you can see your ligand together with the surface of the protein, you can see whether you can expand your ligand a little bit further, or if your idea seems to be too large. Also, we would like to see the atoms of the protein, since the heteroatoms are usually the ones we're aiming at. So we could see whether our new nitrogen is actually close to the oxygen in the protein, which was our intention. So we think those would be quite easy enhancements to add, and I think it's ongoing. Yes.
So going back to where we were before Marvin Live, so we asked ourselves the question how to make better ideas faster. So what about access to the 3D world? Well, as you have seen today, instead of writing 2D papers, anyone in the team can open Marvin Live and very quickly in the sketcher draw a molecule, and then you get access to our local alignment with our own crystal structures. We can align towards the PDB, and also conformers. So very easily you just draw in 2D, and then you have access to the 3D world. Checking ideas. Instead of having to go to Jenny and go back that iteration, as Jenny has shown now, we can, depending on what type of question you're looking at, if it's a property-based design, I want to decrease the pKa, I want to do this and this, or if it's a structure-based idea, you have instant, on-the-fly feedback from Marvin Live. So you can make a better idea, and also you can make many ideas. You can populate your idea with many structures. And then, of course, the improved workflow. Instead of multiple post-its or scribbled piece of paper, Jenny can get SC files, and also that we have the report files that we keep, that in the meetings we can just bring them up and have everything sorted. The tasks are there, so the internal collaboration is much easier to handle. And Jenny wants to add something.
I forgot to say something important. Speaking of faster, I mean, we're doing this very often. So creating a report file is not a very slow or tedious thing to do. Our only problem is that we're doing it so often. So in my current project, the PowerPoint collection of ideas is now like 400 slides. So you can imagine that we're doing this very often. So even though it doesn't save us so many minutes every time we do it, it adds up by the end of the week.
And still, we still like ideas scribbled down on post-its, and we get that still. So it's still the old-fashioned way. But I think this is a way to we talked about having something, we talked about the wow factor during the dinner yesterday, and going into Marvin Live, seeing that something is so simple, so easy to handle. You draw something and then you get access to all this. It's a wow factor, and hopefully, well, I get happy. So hopefully the rest of the team also will get happy and use it more. So we'll see how this goes. So thank you for your attention.
You have access to a number of alignments and X-ray structures. How often does Marvin Live get it right for very similar molecules and for lesser similar molecules?
Yeah, we're still investigating this. And sometimes, as in the case that you saw, you get really nice alignments. And in other cases, it doesn't work. So I have been talking to one of the ChemAxon colleagues about this, and there are ideas on how to improve the alignments. So I think it's, for us, the alignments are key. If you get good alignments, then you get really happy and you can make analogs. As I was also showing in the phenylamide case, sometimes you just totally lose the alignments. And then it's important that the chemists still try to think, could this work? And I would be happy to check the idea on the Linux machine. So, as we said, we want super fast, super good, super cheap alignments. That's important. And I think it could definitely be improved, it sounds like.
This is not my question, this is something that came in on Twitter. Somebody suggests that the CAD people are IT in disguise. You said you had no IT people. You still need to set up email, sharing platforms, etc. Are you IT in disguise?
Well, Fredrik is everything in disguise. But this is not complicated. What we do when we are sharing the files, the SD files, we have access to common folders on my PC. I see the folder that Fredrik sees. And it's not like we don't want to talk to each other when we are now starting to use Marvin Live. I mean, if Fredrik has drawn something and added something in this common folder, he tells me, hey, Jenny, you have something in your mailbox or the folder, and I will have a look at it. So it's not complicated to share the files. Or did I misunderstand the question?
No. This guy is an IT
Oh.
geek, isn't he? And so he's just looking after his job. I'm just repeating it.
Aha. Well,
Well,
we don't need him.
Well, to be fair, of course, we need them, but we don't have the capabilities in-house.
Yeah.
So that's
Yes.
also one of the things that why it's so easy to work with Andras and his team with Marvin Live, because we get all the help that we need.
Yeah.
So-
So they need to create the
Yeah.
iWatch for
Yeah.
us.
Exactly.
Ligand alignments in itself, it's quite a complex kind of operation. Could one envision that the Marvin Live would call an external tool, or maybe like doc or alignment, and then report the results back?
So if you think back on my slides, I mentioned that we have roughly 60 or so plugins right now. Those are like demonstration plugins. So the 3D alignment model that Sprint Bio uses is actually like the ChemAxon alignment. If you open up Marvin Sketch, go to Conformations, you can find the tool there. It's exposed through web services, in JChem Web Services, that's what's connected there. They don't know this because they wanted a solution and we put it together. But the plugin is there, so you are welcome to plug anything in there. And we ship the application with a 3D viewer component that takes the 3D atomic coordinates and displays the results. But anything can feed the data to that. So all the trusted modeling tools have plugin or API or web service components, and those can be connected here. So you're welcome to try your own or evaluate the one in Marvin Sketch.
What are you going to do about reactions? Because you've got exactly the same problem for chemists to say, I want to make this, I'm trying to optimize this reaction. Has anybody got any ideas, and what can you do to help?
So that's kind of a speculative question, so here's a speculative answer. So I would search the IDBS ELN for useful reactions that have a product similar to what I've thrown to see if there's useful information there. But of course you can look at Reaxys or any other data source that has reactions, or take the reaction apart and just look at the product as you have done with the other plugins. So it depends on the scenario. But I think the ELN there is key because it has like the entire knowledge space of the company, so it would be stupid not to use that.
Yeah, my point was more about the collaborative, because it's a fantastic tool for allowing chemists to collaborate. So rather than doing the lookup, and because you know what, we're human, we don't bother to look and search for what people have done before. We'll put it out there and say, has anybody got any ideas on how to help me scale this up or something like that? So exactly what you're doing there is collaborating between a chemist and a computational chemist. It's collaboration between two synthetic chemists or a group of chemists was more my idea there.
Sorry. In that case, the fast and simple answer would be that, so like Marvin.js has very good and improving reaction handling support. So that's there today, right now, and we can see where that takes us. Plugins then would be a different question, so that's a separate topic. But I would build on what Marvin.js has, and you guys know that.